Imagine looking through a microscope and zooming in, deeper and deeper, past the level of human cells, past bacteria, until you reach something almost impossibly small. That is where you find the virus. Viruses are the smallest infectious agents known to science, measuring between 20 and 300 nanometers in diameter. Unlike bacteria, which can be seen with a standard light microscope, viruses are so small that they can only be visualized using an electron microscope. Here is a question that has fascinated scientists for over a century. Are viruses alive? The answer, by most biological definitions, is no, at least not on their own. Outside of a host cell, a virus is completely inert. It cannot generate energy, it cannot grow, it cannot reproduce, it simply exists as a dormant package of genetic information, waiting for a cell to hijack. This is because viruses lack the fundamental machinery of life. They have no ribosomes to make proteins. They have no mitochondria to produce energy. They have no metabolic activity whatsoever. That is why we call them obligate intracellular parasites. The word obligate means they have no choice. They must enter a living cell to do anything at all. Only inside a host cell can a virus commandeer the cellular machinery and begin to replicate. Every living organism on Earth carries both DNA and RNA as part of its genetic machinery. Humans have DNA as their genetic blueprint and use RNA as the messenger that carries instructions to ribosomes. Viruses follow a different rule entirely. Each virus contains only one type of nucleic acid, either DNA or RNA, never both. This is one of the most fundamental and universal principles in virology. Now that we understand what a virus is, let us look at how it is built. At the heart of every virus is its genome, the genetic blueprint, made of either DNA or RNA, that carries all the instructions needed to hijack a host cell and produce new viruses. Surrounding the genome is the capsid, a protective protein shell assembled from repeating subunits called capsomers. Together, the genome and capsid form the nucleocapsid, the core unit of every virus. Some viruses stop there, a naked nucleocapsid, tough and resilient. But others go further, wrapping themselves in a lipid envelope derived from the host cell membrane. Embedded in this envelope are glycoprotein spikes, the virus's tools for recognizing and attaching to new host cells. The capsid is the virus's first line of protection, or its armor. Built from repeating protein subunits called capsomers, the capsid self-assembles with remarkable precision. These proteins fold and lock together through weak non-covalent interactions, forming a structure that is both strong and highly organized. The capsid serves two critical functions. First, it protects the viral genome from degradation, specifically from enzymes called nucleases that would otherwise destroy naked nucleic acid. Second, it determines the shape of the virus. Viruses come in three fundamental architectural forms, each defined by the symmetry of its capsid. The first is icosahedral symmetry, a near spherical shape built from 20 equilateral triangular faces. Many of the most important human viruses, including adenoviruses and herpesviruses, are icosahedral. The second is helical symmetry, a rod-like or filamentous structure where capsomers wind around the genome in a spiral. The influenza virus is the classic example. The third is complex symmetry, seen in viruses like bacteriophages, which have elaborate structures combining icosahedral heads with helical tails, resembling microscopic robots. Not all viruses are naked. Many of the most clinically significant viruses, including HIV, influenza, herpes, and hepatitis B, are wrapped in an additional layer called the envelope. The envelope is a lipid bilayer, and here is what makes it remarkable. It is not made by the virus itself. It is stolen from the host cell membrane as the virus buds out during release. Embedded within this lipid membrane are viral glycoprotein spikes, proteins that protrude outward like antennae. They are the virus's primary tools for recognizing and attaching to specific host cell receptors. They are also the primary targets of the immune system's antibodies. The envelope gives the virus a disguise and a key, but as we will see next, it also gives the virus a critical vulnerability. The envelope is a double-edged sword. On one hand, it provides the virus with sophisticated attachment machinery and a degree of immune camouflage. On the other hand, it makes the virus extraordinarily fragile. Because the envelope is a lipid membrane, it is easily disrupted by heat, drying, detergents, and alcohol. This is why hand sanitizer works against enveloped viruses like influenza and SARS-CoV-2. The alcohol simply dissolves the lipid envelope, destroying the virus instantly. Non-enveloped viruses, by contrast, are tough and resilient. Protected only by their protein capsid, they can survive on surfaces for hours or even days. They resist drying, stomach acid, and many common disinfectants. This difference in stability directly determines how viruses are transmitted, and how we prevent their spread. Non-enveloped viruses are tough. They survive outside the body, in the environment, on surfaces, and in the acidic conditions of the gastrointestinal tract. This makes them perfectly suited for fecal-oral transmission. The route used by poliovirus, norovirus, and hepatitis A enveloped viruses are fragile. They cannot survive long outside a host. They require direct contact, blood, sexual secretions, or respiratory droplets, to pass from person to person. This is why HIV is transmitted through blood and sexual contact, and why influenza spreads through respiratory droplets. We established earlier that every virus carries either DNA or RNA, never both. DNA viruses typically carry double-stranded DNA, a stable, high-fidelity molecule that replicates with the help of proofreading enzymes. This stability means DNA viruses mutate slowly, making them more predictable and, in many cases, easier to target with vaccines that provide long-lasting protection. RNA viruses, by contrast, typically carry single-stranded RNA, a far less stable molecule that replicates without proofreading. Every time an RNA virus copies its genome, errors accumulate. Most of these errors are lethal to the virus, but occasionally, a mutation confers an advantage, resistance to a drug, evasion of an antibody, or increased transmissibility. Within the world of RNA viruses, there is a further critical distinction: the polarity of the RNA strand. Positive sense RNA viruses carry a genome that can function directly as messenger RNA. The moment the genome enters the host cell, it can be immediately read by ribosomes and translated into viral proteins. This gives positive sense RNA viruses a significant head start. They begin producing proteins almost instantly. Negative sense RNA viruses carry a genome that is the mirror image of mRNA. It cannot be directly translated. Before any protein synthesis can occur, the virus must first transcribe its genome into positive sense RNA using a special enzyme it carries within its own capsid, the RNA-dependent RNA polymerase. We now arrive at the heart of virology, the replication cycle. This is the process by which a single virus particle, a single virion, enters a host cell and, within hours, produces thousands of progeny virions ready to infect new cells. The replication cycle begins with attachment, the moment the virus first makes contact with its target cell. Viral attachment is exquisitely specific. Viral surface proteins, the glycoprotein spikes, recognize and bind to particular receptor molecules on the surface of host cells. This lock and key specificity is what determines which cells, tissues, and organs a virus can infect, a property called tropism. Once attached, the virus must cross the cell membrane and enter the cytoplasm. This step, penetration, occurs by one of two mechanisms, depending on whether the virus is enveloped or not. Enveloped viruses enter by membrane fusion. The viral envelope merges directly with the host cell membrane, releasing the nucleocapsid into the cytoplasm. This fusion is mediated by specific viral fusion proteins and often requires a drop in pH to trigger. Non-enveloped viruses enter by endocytosis. The host cell engulfs the virus in a membrane-bound vesicle called an endosome. Once inside, the virus escapes from the endosome, often by disrupting the endosomal membrane, and releases its genome into the cytoplasm. The genome has now entered the cell, but it is still wrapped in its capsid. Before replication can begin, the capsid must be dismantled. This process is called uncoating. Uncoating is triggered by cellular enzymes and the specific chemical environment inside the cell. The capsid proteins are degraded or disassembled, releasing the naked viral genome into the cytoplasm or directing it toward the nucleus. With the genome now free inside the cell, the virus executes its most audacious act. It hijacks the host cell's machinery and transforms it into a viral factory. The host cell's ribosomes are commandeered to translate viral mRNA into two categories of viral proteins. Early proteins, including enzymes like polymerases, are made first and are needed to replicate the viral genome. Late proteins, the structural components of the capsid and envelope, are made afterward and are used to assemble new virions. With viral proteins and replicated genomes now abundant inside the cell, the next step is assembly, the construction of new, complete virions. Assembly is a process of molecular self-organization. Capsomered proteins spontaneously recognize each other and fold together, forming capsid shells. Viral genomes are then packaged into these shells. For enveloped viruses, the process is more complex. Viral glycoproteins are synthesized and inserted into the host cell membrane, marking the sites where new virions will bud. The nucleocapsid migrates to these sites and begins to acquire its envelope. The final step of the replication cycle is release, the exit of new virions from the host cell, ready to infect new targets. Non-enveloped viruses typically exit by lysis. They accumulate inside the cell until the sheer number of virions causes the cell membrane to rupture. The cell dies, and thousands of virions are released simultaneously in a burst. Enveloped viruses typically exit by budding. They push through the host cell membrane, acquiring their lipid envelope in the process. Budding is more gradual and allows the cell to survive longer, producing virions continuously over an extended period. We have seen how viruses are built and how they replicate. Now we turn to the question that matters most in clinical medicine. How do viruses cause disease? Every viral infection begins with a portal of entry, the site through which the virus first gains access to the body. The respiratory tract is the most common portal. Viruses enter through inhaled droplets or aerosols and attach to the epithelial cells lining the airways. The gastrointestinal tract is the portal for fecal-oral viruses. These viruses are swallowed, survive the acid of the stomach, and infect the intestinal epithelium. The skin provides a portal when its integrity is broken, through wounds, insect bites, or needles. The genital tract is the portal for sexually transmitted viruses. The portal of entry is not just a starting point. It determines the initial site of infection, the pattern of spread, and the clinical presentation that follows. One of the most clinically significant routes of viral transmission is vertical transmission, the passage of a virus from a mother to her child. This can occur at 3 distinct time points, each with different consequences. Transplacental transmission occurs during pregnancy, when the virus crosses the placenta and infects the developing fetus. This is the most dangerous route, as it can cause severe congenital abnormalities. Intrapartum transmission occurs during delivery, when the newborn is exposed to maternal blood and secretions in the birth canal. Caesarean section can reduce this risk for some infections. Postpartum transmission occurs after birth, primarily through breastfeeding. Not all viruses spread beyond their initial site of entry. Some are content to remain localized, replicating exclusively in the tissue where they first landed. Local infections are generally less severe than systemic infections. The immune response can be mounted quickly at the site of infection, and the damage is confined to a limited area of tissue. Other viruses do not stay local. After initial replication at the portal of entry, they enter the lymphatic system or bloodstream and spread throughout the body, a condition called viremia. Viremia can be primary, the initial spread from the entry site, or secondary, a second wave of viremia after replication in internal organs. Secondary viremia typically produces higher viral loads and more severe disease. Systemic infections affect multiple organs simultaneously, explaining the widespread symptoms of these viral diseases. Viruses cause disease by damaging cells, but the mechanisms of that damage are more varied and more sophisticated than simple destruction. When virologists grow viruses in cell cultures, they can observe the damage directly under the microscope. The visible changes that infected cells undergo are collectively called the cytopathic effect, or CPE. CPE takes several forms. Rounding and detachment is the most common. Infected cells lose their normal flat shape, become spherical, and peel away from the culture dish. This reflects disruption of the cytoskeleton and cell adhesion molecules. Inclusion bodies are another hallmark of CPE. Dense aggregates of viral proteins or viral replication complexes that accumulate inside the nucleus or cytoplasm. Some inclusion bodies are so characteristic that they serve as diagnostic markers. Finally, lysis, the rupture and death of the infected cell, is the ultimate cytopathic effect, releasing new virions and triggering local inflammation. Some viruses have evolved a remarkable strategy for spreading from cell to cell while evading the immune system. They cause infected cells to fuse with their neighbors, creating massive, multinucleated structures called syncytia, or giant cells. The mechanism involves viral fusion proteins expressed on the surface of infected cells. These proteins interact with receptors on adjacent uninfected cells, triggering membrane fusion. The result is a single enormous cell containing multiple nuclei, a syncytium. By spreading through cell fusion rather than releasing free virions, the virus can move from cell to cell without ever entering the extracellular space, where it would be exposed to neutralizing antibodies and complement proteins. Perhaps the most alarming long-term consequence of viral infection is malignant transformation, the conversion of a normal cell into a cancer cell. Approximately 15 to 20% of all human cancers are attributable to viral infections. Certain viruses have evolved mechanisms that directly interfere with the genetic controls governing cell growth and division. The first mechanism is the inactivation of tumor suppressors, as p53 and retinoblastoma protein, two of the cell's most critical brakes on uncontrolled division. Remove the brakes, and the cell proliferates without restraint. The second mechanism is chronic inflammation, causing repeated cycles of cell death and regeneration. Each cycle of regeneration carries the risk of DNA replication errors. Over time, these errors accumulate into the mutations that drive tissue cancer. The third mechanism is insertional mutagenesis. Retroviruses integrate their DNA into the host genome. If integration disrupts a tumor suppressor gene or activates an oncogene, malignant transformation can follow. We have followed the virus from its first contact with the body through its replication, spread, and cellular damage. Now we arrive at the final question: how does the story end? Let us examine each of the three possible outcomes in turn. The first is subclinical infection, the silent infection. The virus enters the body, replicates, and is eventually cleared by the immune system, all without the host ever experiencing a single symptom. This is far more common than most people realize. The infected individual develops immunity without ever knowing they were infected, but they may still transmit the virus to others. The second outcome is acute infection, the classic pattern of viral disease. Symptoms appear, often abruptly, as the immune system mounts its response. The battle is intense but brief. Within days to weeks, the immune system prevails, the virus is cleared, and the host recovers, typically with long-lasting immunity. The third outcome is persistent infection. The virus is not cleared. It remains in the body for an extended period, sometimes for life. This is the most clinically complex outcome. Persistent infections are not a single entity. They fall into three distinct categories, each with its own biology and clinical implications. Chronic infection is characterized by continuous viral replication at a detectable level. The virus is always present, always replicating, always detectable in blood or tissue. Latent infection is more subtle. The virus establishes itself in a specific cell type, often neurons or lymphocytes, and enters a dormant state. Viral replication ceases. The virus becomes undetectable, but the viral genome persists, silently, within the host cell's nucleus. Periodically, triggered by stress, immunosuppression, or other stimuli, the virus reactivates, resuming replication and causing recurrent disease. Slow infection is the most insidious. The virus replicates at an extremely low level for years or even decades, causing a gradual, progressive, and ultimately fatal disease. We have now completed our tour of the viral world, from the nanometer-scale architecture of the virion to the decades-long consequences of persistent infection. And we return to the question we began with: how can something so microscopically small be so enormously powerful? The answer lies in evolutionary precision. Viruses have had billions of years to refine their strategies. They carry only what they need, a genome and a protein code, and they rely entirely on the extraordinary complexity of the host cell to do the rest.